A system of transport called the phosphotransferase system, or designated as PTS, uses the energy from phosphoenol pyruvate to transport sugars such as glucose into the cell. PEP is an intermediate in glycolysis and carries a high energy phosphate group. In this system, the phosphate group of PEP is transferred to the cytoplasmic enzyme called enzyme 1. In turn, enzyme 1 transfers the high energy phosphate group to a histidine-rich protein called HPR. HPR then passes the phosphate group to the transport protein designated as enzyme 2, which consists of three domains A, B, and C. After enzyme 2A receives the phosphate group, it transfers the phosphate group to 2B. Enzyme 2B delivers the phosphate to the glucose, which is transported into the cell by enzyme 2C. In receiving a phosphate group, glucose is converted into glucose 6-phosphate. More than 20 different types of carbohydrates, including mannose and mannitol, can be transported by the phosphotransferase system. The system has a modular design, with enzyme 1 and HPR being common to all of the sugar transport system, but however the enzyme 2 is substrate specific. Mannitol and mannose uses unique enzymes too for transport into the cell, in the process becoming mannitol 6-phosphate and mannose 6-phosphate respectively. Note that the phosphotransferase system is a type of active transport because it uses the energy in the form of a high energy phosphate group to chemically alter the solute. Because the modified carbohydrate inside the cell is a different chemical entity that is related to the compound outside, the parent solute entering the cell is always moving down its concentration gradient, regardless of how much solute has been already transported.